Good morning, everyone, or good afternoon, depending on where you are joining us from today. We'd like to welcome you and thank you for attending the second day of our webinars entitled Validating, Optimizing, and Monitoring EO Sterilization. During this webinar, we will focus on validated cycles and cover annual requalification, annual review activities, first steps to cycle optimization, and regulatory considerations. Just a few housekeeping items we'd like to take care of. If you miss any of one of our webinars, or you have to refer back to this one or the day one session or any other webinar, you can always find them on the Sterilization or Nelson Labs website in the resources or education pages about a week after the live event. On our websites, you also find information about upcoming seminars where we dive deeper into many different topics. You can find brochures, videos, and also white papers. Also, follow us on LinkedIn to learn about more upcoming educational opportunities. As for today's webinar, we welcome your questions. You can submit them at any time throughout the webinar using the question button. Our presenters will try to answer as many as time permits during the live Q&A at the end of their presentation. The remainder will be responded to individually after the webinar. If you have any product-specific question, please submit that and our presenter or account manager will be able to respond to you individually after the event. So I'd like to now introduce today's presenters, Russ Gonczarowski and Mike Padilla from Sterilization. Russ is a longtime employee of Sterilization, having held roles as an EO consultant, EO validation coordinator, microbiology technician, and microbiology laboratory supervisor. Today, Russ is a technical advisor who leads ethylene oxide sterilization projects in numerous facilities across North America. Russ enjoys sharing his knowledge and often gives training sessions on EO sterilization to customers and notified bodies. In his role as Technical Director US of Sterilization Expert Advisors, Mike Padilla is responsible for leading the EO Expert Advisors team in the development and management of ISO and FDA compliant sterilization studies. Prior to joining Sterilization, Mike held positions with Baxter Healthcare and Allegiance Healthcare, where he managed various projects ranging from product development to validations in steam and EO sterilization modalities. Mike has been published in medical design and outsourcing, and also is a member of the AMI Sterilization Committee Group, Working Group 1, Industrial EO Sterilization. We'd like to now welcome Russ and Mike, and I turn the time over to them.
Thank you, Mike. Good afternoon, everyone. Just to identify myself, this is Russ Gonczarowski. I'm going to start off, and then I'm going to pass over to my colleague, Mike. The slide you see here is just a summary. Perhaps you were able to attend session 1, where Courtney and Dania gave you some very useful information. Day 2, this session, I'm going to talk about, as Mike mentioned, annual review activities as they apply to cycle optimization. I'm going to go over the annual requalification process a little bit, and I'm going to turn it over to Mike, and he's going to talk about cycle optimization and some regulatory considerations. So we'll start first with the annual requalification review. I'm going to talk about this as it applies to ISO 11135, the requirements and guidance surrounding that requalification review. I'm going to talk about what we need to look at as far as the revalidation review, including the equipment, the product, and the processing history, and some details there. Then I'm going to go over some of the outputs of that review and apply them to some bigger concepts. So requalification. Performing an annual requalification review is a requirement to maintain your validation process, but it can be used for a lot more. The purpose of the review is to collect data, gather information, investigate, and review your process. And it can be a great opportunity to evaluate your sterilization process and identify ways to enhance the efficiency. This can be done to continue the safe usage of EO in order to lower your residuals from the process to perform better than the patient safety regulations dictate. It could allow for reduction in required aeration time, resulting in an improved customer supply chain efficiency, a faster turnaround time, and it may reduce your cycle time, resulting in additional capacity for manufacturers for the sterilization process as it applies to EO. Looking at the standard ISO 11135 itself, Section 12 covers maintaining the process effectiveness, and specifically Section 12.3 talks about requalification. And what it says is that the IQ, OQ, PQ, and subsequent requalifications shall be reviewed annually. So that's a requirement to determine the extent of requalification that is necessary. This includes an assessment of the need to confirm the product's sterility assurance level through microbiological studies. The outcome of this review, including the rationale of decisions, should always be documented. Requalification of the process carried out with specific equipment shall be performed at defined intervals against specified acceptance criteria and in accordance with your documented procedures. These intervals shall be justified. So what this means is the simplest route to compliance is to have a documented procedure or work instruction that requires an annual review of the validation. And it should include IQ, OQ, and PQ. And the guidance in Annex D, in that section D.12.3.2, is a very useful tool in establishing a requalification process. We'll take a look at it. So this figure, taken directly from 11135, is the requalification decision tree, and it basically leads you through a pathway of what you need to do at which time and the decisions that need to be made during that process. So I'll highlight specifically this information on the left side of your screen for the annual review. What we want to review is, first of all, the equipment, secondly, the product, and lastly, the processing history. And we'll go over that in a little bit more detail. Even where elements of the sterilization process are contracted out, it's important to note that the medical device manufacturer is ultimately responsible for the validation, release, and distribution of sterilized product to the market. Therefore, the phases of this annual review are the manufacturer's responsibility. However, this presents a great opportunity to partner with your contract sterilizer to obtain all the information that's necessary for the process during the annual review. The first phase of the annual review is to evaluate how the equipment has been performing. So firstly, we're going to look at changes to the equipment and determine, have there been any changes to the equipment? Have the changes been requalified, and has that documentation been provided? The second phase of the equipment review should cover any calibration. This would absolutely include a review of the installation qualification of the chamber or other monitoring equipment. And it includes any calibration of the control and monitoring equipment associated with the equipment. Next, a review of the OQ status should be performed. Has a periodic requalification of the equipment been performed? How does that affect the process? Lastly, for the equipment review, you would want to evaluate any maintenance performed, whether it's preventative or due to a specific event. Was the equipment appropriately requalified after any maintenance intervention, and has that maintenance been properly documented? Once again, this is a great opportunity to work with a contract sterilizer to attain this information because they would have work instructions or procedures in place in order to evaluate the equipment on an annual basis. The next phase of the annual requalification review would involve the product. Any changes to the product or package should be evaluated and documented accordingly. So when we're talking about the product, first we would take a look at the product design and have there been any significant changes to the product design, perhaps due to any feedback from the field or the end users. Also with the product design, you would want to evaluate if any new products have been adopted into the validation process and what products those product families, what product families those products may fall under. The next phase of the product review would involve the packaging and to determine if there's been any significant changes to the packaging. Some quick examples there would be a change to the instructions for use, or IFU, a change to the breathable area, in other words, the Tyvek portion of any primary packaging, or a change in any other packaging materials that are utilized. Speaking of materials, that brings us into the next phase of review as it applies to the product. And have there been any significant changes to the materials? This would involve any new suppliers or new vendors that are providing any material during the manufacturing process. Once again, part of the product review would be any maintenance. And this would be, has there been any significant changes in the manufacturing process equipment that it could impact the product characteristics? Examples of this include a change to the bioburden, the EO residues after the process, and the sterility assurance level of the product. Speaking of bioburden, has the bioburden test been performed in accordance with ISO 11737-1? Is the bioburden data consistent with the original validation after monitored or trended? Lastly, for the review, the load configuration should be reviewed. And have there been any significant changes to the load configuration? This would include any repackaging of the shipper cartons, any different layers added to the load as it's palletized, any change in density, increase or decrease, if there are more or less products placed on the package within the packaging, or any change to stretch wrap, banding, or corner boards that's presented for the load configuration during transport. The last phase of the requalification review involves the process history. Are there any process design changes to the sterilization process? For example, is there a change to the process temperature, any change to the steam addition phases, or the gas exposure time, or any evacuation changes? The next portion of that review would be any process deviations, specifically any biological indicator failures identified since the previous requalification. We'll talk about that a little bit more in depth later. Next, we would look at any process failures of the equipment, and if there are any nonconformances noted due to failure or malfunction of the equipment. Next, if the process is a parametric release process, some processes are designed to only look at the parameters and eliminate the BI sterility step, a capability assessment should be performed to evaluate key parameters, specifically the humidity and the EO concentration. Lastly, what should be reviewed are any other process history. That will include the number of processes performed since the previous requalification, a capability assessment of the process parameters, and perhaps the ethylene oxide residues that are left over after the EO process. Okay, now that we've gone over several different phases of the annual requalification review, we're going to provide some examples of what an output of that review would be. And these are, again, taken directly from that decision tree in the standard ISO 11135. So the first example is scenario 1. After the review, maybe you've identified that there are some changes to the product or process, but they're not significant. You have a good processing history since the last requalification, and the previous qualification was performed 1 year ago. Once again, this would be no changes to the product, packaging, equipment, or process, and there's an acceptable chamber performance and engineering review. The sterilization process is operated reliably within the review period. Well, then no physical or microbiological PQ would be required. The only thing that would be required in this scenario would be documentation of the review and documentation of the rationale, the output of a review, that no physical or microbiological PQ is required. In other words, there are no changes identified. Therefore, we do not need to perform a physical study. Next, we have scenario 2. This would be another output of the annual review. Maybe there are some changes, but they're not significant, and there is a good processing history. And the validation or last requalification was performed 2 years ago. This would identify a need to verify the continued appropriateness of the resistance of the internal PCD to the product Bioburden. After a defined interval, this would provide evidence that there has been no inadvertent changes since the previous qualification study. In this case, in this scenario, a reduced MPQ or PPQ may be required. This would typically include 1 fractional or half cycle exposure. Within that 1 fractional or half cycle, what should be included is a low temperature and humidity measurement. It is recommended that a reduced MPQ half cycle physical study with the low temperature and humidity measurement be performed at least every 2 years to verify that the documented paperwork review has captured any inadvertent changes. Finally, we'll go into a third example, a third scenario. If during the review you've identified some significant changes and there is a good processing history, or you've identified that there are no changes, but there's a very poor processing history, this might have identified a significant change to the product packaging design or configuration, the process design, or even the equipment or service. In this scenario, it would be our worst case scenario because we'd have to perform a full qualification, including an MPQ and PPQ. In other words, going back to the drawing board to start the validation again. Once again, this can be required in certain situations following a significant change. If you have identified a significant change, some examples of such a change discovered during the review are provided below, and this is certainly not a complete list. So some examples of this would be a BI sterility failure, a need to observe lower residuals that perform better than the patient safety regulations as they exist, a bioburden data shift, or process history nonconformances or deviation. So what I'm going to do next is go through a couple of these examples and identify areas of review or questions that need to be asked if one of these situations occur and a significant change is identified with one of these examples. It's important to understand the why behind these significant outcomes. So for a BI sterility failure, what you would want to ask is the process challenge device an accurate measurement of the resistance to the EO process? Is the external PCD appropriately documented? Are the cycle parameters sufficient to achieve the required SAL? Excuse me. Have there been any inadvertent changes to the product, materials, packaging, or load configuration? Sometimes those small changes can add up. What you would also want to do is review the documented data for BI appropriateness to identify areas where the internal and external PCDs can be redesigned if they were originally designed around a higher bioburden resistance. We'll get more into bioburden in the next slide. You want to review the original EPC data to understand if it may be too resistant compared to that of the qualified internal PCD. Is the EPCD resistance forcing an extended cycle length? The next output of the review may be a high residual level. And some of the questions that need to be investigated or asked about this follow. Have new materials been introduced which would cause the device to retain a higher gas residual level? Has there been a switch to a new supplier or vendor due to cost concerns? Has there been a packaging or load configuration change due to transportation issues? This would be to and from the contract sterilizer, at the sterilizer itself, and then transportation to perhaps the distribution warehouse or the end of where the product needs to go. Has there been a change to the aeration equipment, including the temperature, the exposure time or dwell time, or forced heated air exchanges? That would have an impact on product residues. Has there been a change to the process parameters, resulting in a higher EO concentration or especially a longer exposure period? Is there an opportunity here to increase the process temperature or the number of gas flushes at the back end of a cycle, which would be designed to remove EO from the product within the chamber? Lastly, I would suggest performing a regulatory review of EO residue testing limits to confirm that your products are properly classified for patient contact under the standard ISO 10993-7. Next, we'll go over the product bioburden control. This should be evaluated on a quarterly basis at minimum. You would want to review the historical product bioburden data to understand how challenging the manufacturing process is to maintain that level or find ways to improve that data. Has a new manufacturing facility been opened in a different geographic location? That would have an impact on the product bioburden. Have there been any major changes in personnel or procedures since the last requalification? Has a higher number of microorganisms been identified exceeding the action or alert levels as they've been established? Have any of the organisms been identified with a higher resistance to the EO sterilization process? Lastly, the output of the review may identify that there's a high number of process history nonconformances or deviations. Some questions to ask about this are, has the routine process consistently met the sterilization process specification? Especially, have any assignable causes been identified for any failures? Has there been a shift in product temperature or humidity observed during any physical requalification PPQ? Has the equipment been able to consistently meet the validated specifications for pressure, time, humidity, or rate? Any of these discussed example cases could present opportunities to redefine your sterilization process. As mentioned earlier, it's important to document the decisions taken and the rationale behind those decisions to keep with your validation master file. Right now, I'm going to turn it over to my colleague, Mike, who's going to talk about the first steps to cycle optimization, but I will be around to answer any questions at the end of the presentation.
Thank you very much, Russ. So what I'll cover on the second half of the presentation that we're focused on cycle optimization is to kind of get you started with the first steps. How do we get started with any project work and so forth? And then also, more importantly, as you're venturing into cycle optimization, regulatory considerations. As we all know, we are in a very tightly regulated industry. We have to follow industry procedures, regulatory procedures, ISO compliance, and so forth. So it's important to, as you're optimizing the process that you've validated, is to also keep an eye on the fact that you may have to update your regulatory department, contact your notified body to figure out what needs to be addressed if there's any gaps that become apparent. So we'll start off with the first steps for cycle optimization. So where are we at the moment? Russ did a great job in terms of walking you through the decision tree that is in the ISO 11135 standard. That decision tree is a flow chart that is the starting point for you to be able to review your process. You've conducted a documented review of your validated process and identified opportunities for optimizing your process. Okay, this is great. But we always want to make sure that we can pause just for a minute and do some homework. If you feel that you need to adjust the temperature of your sterilization process and raise it, or you find opportunities in the pressure or humidity characteristics of your process, you want to take time and look at the historical data that you have on file. Again, we're dealing with a validated process, so you have a goldmine of information available at your fingertips, hopefully, that you can easily point to in terms of looking at what did you test for the products and for the packaging as part of your original validation. And then use that information to identify if there's any limiting factors that may have just been put into place with your current process. You want to assess if the legacy data is still relevant to the current product packaging. Are there newer improved materials available today that were not available before that could be considered as alternatives? Or will you conduct supplemental product package testing with the optimized parameters? So this homework is very valuable to understand how far you can go with any further discussion on proposed optimization plans. Here are some examples of key areas you might want to look for optimize. Similar with some of the areas that Russ talked about, you may be in a position that with your validated process, you may want to reduce cycle length in efforts to perhaps process more product through the chamber. Makes perfect sense. You may also look at reducing the amount of EO used in your cycle, and also improve on the EO residue levels in your product. We're in a situation today in the environment of the EO industry where the industry as a whole is now looking to optimize or make cycles more efficient with respect to utilizing ethylene oxide. And I'll talk more about this later on. But this is a very interesting opportunity in terms of bullet point number 2. The third example I'll talk about is addressing BI failures. BI failures with respect to routine processing. Now, looking at reducing cycle length, if I was in a conversation today with a medical device manufacturer looking at what can we do for reducing cycle length, what are the opportunities, what should we be considering? This is a list, not comprehensive at all, but it does help you point out some of the areas that I would start looking into with you and the product and packaging. And I want to highlight a few of them that I think are perhaps the first areas that I would start looking into. Looking at the temperature of your sterilization process. If I see today that the temperature of your process is not higher than I would like, I would always encourage to look at increasing the temperature of the process. As you increase the temperature of the process, you have a chance to improve the effectiveness of the ethylene oxide molecule, not just for sterilization, but for degassing. So consider looking at that. And also pointing out further down the list, the opportunity today with the fact that the FDA issued in 2019 a innovation challenge to look at the medical device industry, to look at utilizing existing alternative approaches for validating the process instead of the traditional half-cycle approach. There are several alternative approaches where one of the benefits would be to potentially reduce the overall cycle time by reducing the gas exposure time necessary to sterilize your product. Don't get me wrong, in the end, these alternative approaches will still provide you to deliver a sterilization process, delivering the minimum sterility assurance level required for your sterility claim. It's just different tools in your toolbox. Other items in this list that would help you in terms of considering cycle length reduction would be like changing the load configuration to decrease the challenge of penetration for EO to sterilize the most challenging areas of that load. If it's density, if it's load configuration, such as how much product or how the boxes are configured or oriented on the load, that's what we mean by load configuration. Can the product and packaging withstand faster program rates? Okay, what this does is it will help conduct the same amount of phases in your sterilization process, but just look at, see if we can shave off minutes in each of the phases. Cumulatively, you would be able to reduce cycle length. Can the packaging be redesigned to improve EO penetration and EO degassing? One example in this is, don't just look at the packaging of the product, but the packaging of the load. And what I mean by that is looking at how we use stretch wrap, or the traditional film that gets used to wrap the pallets. Although used as a way to restrain the load on the pallet, it can also be a barrier that you have to deal with in terms of getting ethylene oxide through that film before it even starts penetrating through your corrugated shipper boxes, and eventually work through the load into the hardest to locate BIs in your product. You can eliminate stretch red film and allow more efficient penetration and degassing the VO sterilant. Second bullet point is reducing the amount of VO used in EO residue levels in the product. This is probably the most popular target that we've seen in the industry over the last 6 to 9 months. Obviously, to accomplish this, I would be focusing primarily on reducing the amount of VO needed to sterilize the load. Traditionally, we've seen gas concentrations for the cycle be somewhere in the range of 500 milligrams per liter up to 700. But today, in the industry, we've seen success even at the lower gas concentrations of 250 milligrams per liter. Great improvement and a significant reduction in EO utilization. We're accomplishing more with using less EO. That's the message I want to send to you, is that we've done innovative things with being able to use less EO per pallet in terms of pounds and still accomplish sterility of the load. The last point on this slide is to perform a regulatory review as well. When I talk about residue levels in the products, one piece of advice I always give medical device manufacturers is to do yourself proper step of looking at the product, and don't just rely on historical efforts that were made in terms of classifying your device. There may be an opportunity that if you review what's been done in the past, you may be in a situation where you can vision in terms of how the regulations are today. For example, look at how the device was classified in the past and make sure that it's relevant today in accordance with ISO 10993-7. If you change the classification, it may, for example, change the limits that you have to meet. And probably in a best case scenario, you may have had too stringent of limits in the past and be able to readdress your limit for the current regulations. On this slide, additional things I wanted to mention is that you can, in order to reduce the amount of VO used and residue levels in the product, you could go down the similar pathway that I mentioned a couple of slides back of areas to look at for cycle length reduction, and apply those same approaches for reducing EO use and residue levels, such as the increase in the process temperature. If we're looking to reduce residue levels in the product, we could easily just say, you know what, just extend heated aeration, but capacity becomes an issue as well. So in the chamber, we would still introduce the same question of, can we increase the process temperature in the chamber? And give ourselves an opportunity for the first step forward of dealing with the reduction of residues in the chamber before we get into heated aeration. So the rest of these items on this list are the same ones that we talked about in the earlier slide for cycle length reduction, but as you can see, there's a lot of overlap. Third bullet point is if you were looking to address BI failures. Russ touched base on this one as well, primarily looking to see if, first ask yourself if the EPCD or the external process challenge device, when it was first developed for your process, that it may have been too resistant compared to that of the qualified IPCD. And is the external PCD causing the cycling to be longer than what is actually required to sterilize products? For example, that in order to sterilize your product from an IPCD perspective, where you're killing the BI in your load, it may require 2 hours of gas exposure, but because your EPCD is much more resistant, you may be requiring 2.5 hours or 3 hours. So that's just something to look at in terms of the EPCD historical data. So if you're having problems with BI failures, we always say look back on the historical data that you have on file for how that EPCD was generated. And recently in my conversations with some customers, I'm asking them, take a look at the historical product Bioburden data. When you establish the hierarchy of resistance between the Bioburden on the product and the resistance of the BI and the IPCD versus the resistance of the BI and the external PCD, that 3-layer hierarchy of resistances is important to understand which did back then with the original validation. So, for example, if the product Bioburden back then is much different today, meaning that you've improved it, look into that, and that might give you an opportunity to improve on the design of the IPCD and EPCD, because you have much better control today of the Bioburden, and that would be the first recommendation. And so you would always want to start there and see if there's room to work with in terms of reestablishing that EPCD so that's more realistic to your products. Now, having gone through those examples, what I want to briefly touch on is regulatory considerations. As you're making these changes, what must you communicate to the regulatory bodies? First of all, don't be discouraged to optimize your process, thinking that, oh, if we change parameters, we got to go through the whole process, it's going to take time, it's going to be a lot of lengthy conversations and so forth. What I want to mention is don't be discouraged. Check with your regulatory agency and your notified body. Be proactive. Once you confirm that there is an opportunity for improvements to any parameters, you should engage your notified body or regulatory agency and let them know of what your anticipated changes will be. The FDA, based on the innovation challenge that I mentioned from 2019, the FDA is engaged with the industry to help push along ways to innovate the sterilization processes. So if you're looking to optimize your process as part of the innovation challenge on how to reduce ethylene oxide use, the FDA is already anticipating 510K submissions to be submitted in regards to this. So I think this is a really good opportunity for us as the industry. As you anticipate changes to be made to your process parameters and you lay out the plan for your validation of this new process, notify the agency and determine what supplemental data will be needed for your 510K submission file. Understanding how these process changes will impact your original submission file is important. Most importantly, how much extra data will you need, if any, with your changes? And it may be specific data, but you want to know this up front so that you're not blindly going through the process anticipating that what you're doing may be appropriate. If your guess is right, great, but it would avoid you a lot of headache down the road by just simply engaging with your notified body and regulatory agency up front. And lastly, different regulators may have other policies, and it's possible that in other countries, more specific data may be required. So inquire. On this slide, I just want to briefly touch on what a 510K is, just for the general public, and its importance. A 510K is required by the FDA to sell medium risk medical devices in the US. A 510K contains detailed technical, safety, and performance information about a medical device. if you are planning to manufacture or distribute a medical device within the U.S., the FDA requires in most cases that you complete either a 510K submission or obtain pre-market approval for your medical device. Here is a list of just some elements of what a 510K incorporates that you will see are capturing information about your sterilization process, such as description of the sterilization method or the modality that you're using, description of the chamber that you're planning to validate, where is that chamber located, description of the method used to validate the sterilization process. This is an important point I wanted to highlight, whereas Russ touched base on it, and also early on day one of the webinar when Daniel Cortez mentioned about the alternative methods for validating. This is an important point where you would talk about the alternative validation method you would be choosing, such as cycle calculation, or if it's a BI Bioburden method. And then obviously the sterility assurance level that you plan for your process to deliver is important, and also a description of the packaging. There are other elements to the 510K, but these are ones I wanted to highlight. And lastly, as I mentioned, in 2019, the FDA announced 2 public innovation challenges to encourage the development of new approaches to medical device sterilization. Challenge number 2 was focused on developing strategies to reduce ethylene oxide emissions. So this challenge 2 is part of the foundation of where the industry is going with improvements on cycle design. And you can read more about this innovation challenge through the FDA.gov website. One important piece I want to make is regarding this challenge. This challenge is not a company-specific challenge. It's an industry-wide challenge, and a lot of the industry is fully engaged in this in terms of redesigning cycles, pushing boundaries of using less ethylene oxide than what we ever imagined compared to what was done 20, 30 years ago. So a lot of success has been done. There's more to be had. And again, this FDA website is available if you want to read more about it. So with that, I thank everyone, and we can take some questions. So
thank
Mike,
you both. Yeah, no, thank you. Thank you, Russ. Thank you, Mike, for all of your thoughts and your insights in this presentation today. We've all learned some valuable information. And you're right, we do have some time for take some questions. So let's just get straight into the questions. And here's the first one. How often am I required to perform a physical revalidation cycle to assess the sterility assurance level of the process?
Mike, this is Russ. I'll answer that one. So for a requalification, or for a physical requalification, the wording in the standard ISO 11135 states to do it every 2 years. So that would be the interval. And this is in order to provide evidence that there's no inadvertent changes since the previous requalification. When I say inadvertent changes, there may be very small changes to either the packaging or the product. Maybe you're utilizing a new vendor or supplier for one of the materials. Maybe you make a small change in the IFU. Maybe the parameter changed slightly. So those individual examples aren't significant on their own, but they could add up over time and stack on each other to bring you out of compliance with your sterility assurance level. So every 2 years is the recommended period to perform a physical requalification cycle. And a reminder that requalification cycle should include measurement of the temperature and humidity profile of the load and compared back to the original PPQ.
Great, thank you, Russ. And here's our next question. What do I need to do if my bioburden levels are trending higher than normal?
So this is Russ. I'll handle that question as well. So the first thing is, not to panic. Bioburden, the reason that we trend that data is because it fluctuates high and low over the course of a yearly period, let's say. So first thing I would recommend is to evaluate any environmental controls within the manufacturing facility and maybe assess any additional training to see if there's any major personnel changes or something of that nature. If you're trending your bioburden and it becomes too high, we know that for ethylene oxide sterilization, we're not directly setting the dose or the exposure based on that bioburden. We've established that we're doing it based on a process challenge device. So I wouldn't be too worried about a high bioburden level. But if it does get to be up to a worrisome level, let's say, I would recommend performing or reconfirming the BI appropriateness in a sublethal or a fractional cycle to compare that process challenge device and make sure that it's more resistant than the natural product bioburden through a product sterility test.
Great. Thank you again, Russ. Okay, on to our next question. How can I evaluate effect on my product if making any changes to cycle parameters?
Another good question. I'll field that. So the best way to evaluate any parameter changes is to perform what would be considered an engineering or a feasibility cycle run. And that would be just simply changing the parameters to what you feel the changes are going to be, whether it's a temperature change or a pressure change, maybe a humidity change, any aspect of the cycle parameters, and performing that cycle on an identified number of products that once the process is done can be evaluated, whether that's a visual inspection or maybe there's some existing product functionality or package integrity testing that was established for the process during the initial PQ. But that short sort of one-off cycle engineering run, that would be what I would recommend if you're changing parameters, just to evaluate what effect those changes have on the product itself before you look into any sterility concerns.
Okay, thank you. We have some time for a few more questions. Do I need to redo any product functionality or package integrity testing if I change my validated method in order to optimize the cycle?
Yeah, Mike, this is Mike. I'll take that question. It's a good question. I think the most important part of what a company should do is to do a documented assessment while looking at the historical data that's available for what was done before on the product functionality and package integrity, and look to see if there's any gaps in the original testing that need to be addressed with you changing parameters. For example, if the temperature is changing, if you're going to be raising the temperature of the process, what effect does that have on the product? If you're talking about increasing the rates, the program rates of the cycle, how will that affect the existing package integrity data? If there are any changes in humidity levels, such as you want to improve the humidity level in the process, how would that need to be addressed in terms of product functionality and package integrity, and maybe even package seal strength for your final packaging?
Thanks, Mike. Hey, on to the next question. What specific information do I need to provide to the FDA if I'm submitting a 510K in order to optimize the validated process?
Another good question. I think if I can stress one thing, there won't be a one answer for everyone. It's important to communicate with the FDA and for the FDA to give guidance in terms of what information they would require specifically for your product. This may include information about the sterilization site or the equipment or chamber or the sterilization method that you're going to be using. So I think the bottom line is reach out to the FDA, pose the situation, and have them look at your file and provide guidance and start there.
Okay, thanks, Mike. Here's our next question. Will cycle optimization allow me to skip any requirements in ISO 11135?
I'll take that question. Can cycle optimization allow me to skip? Absolutely not. Great question. I think what it comes down to is cycle optimization is its concept. You're dealing with a validated process, but then through cycle optimization, you'll eventually be changing parameters because that's the essence of cycle optimization. Can I improve this? Can I shorten this? Can I make this more aggressive than it was because my product or my packaging can withstand a much more aggressive cycle today? The bottom line is that you still have to comply with all the sections of the ISO 11135 standard to be able to show evidence that you have a validated process. Keep in mind that you had a validated process in the beginning. You're going to be changing parameters to optimize them. So in essence, you have a different process. So you'll have to go through 11135 and show evidence that you're complying with all the important sections, such as process definition in Section 8, and obviously the MPQ and PPQ validation that Russ talked about in Section 9. Great question.
Okay, thanks again, Mike. I think we have time for one last question. If I have more than 2 chambers qualified to the same cycle, can I reduce the requalification to only 1 chamber representing all?
Good question, Mike. This is Russ. So I'll field that question. The short answer is yes. Really, within the standard, there's a section regarding process equivalence, and that's making sure that cycles that are qualified to separate chambers, that the cycle parameters are operating in the same way from a statistical analysis standpoint in each of the chambers. So if you perform that process equivalence in each of the 2 chambers qualified, and you've evaluated that, then that could allow you to reduce the number of requalification runs for only 1 chamber. However, if you've not done that process equivalence, assessment, then I would recommend still performing a requalification reduce in each chamber. But that statistical analysis of the process equivalence, that's key to reducing the number.
Great, thank you, Russ. And unfortunately, we are out of time today. And we'd like to thank everyone for attending this webinar. And we want to thank again Russ and Mike for sharing their expertise with us. As a reminder, you can access all of our previous webinars on the Sterigenics.com or NelsonLabs.com websites under the Education or Resource tabs. And you'll be able to access the recording of this webinar in about a week after this event. So we also like to, again, thank you for attending. We hope you have a great rest of your day.